Good morning, my beautiful brothers and sisters. I pray you're all having an amazing day. I'm going to try to keep this as short and sweet as possible because I'm going to actually tag this onto the video that you see on your screen right now. And I know a lot of it is, it's a lot of science mumbo jumbo that I don't understand 3/4 of it, to be honest with you. But I know a lot of you out there definitely can. I know, a lot of you have had experience in this field, whether it be within medicine. I know our brother, David, Jesus Freak Computer Geek, working for NASA. And a lot of people have different skill sets that, being a part of the body of Christ, we're all different and bring different things to the table. But the one thing that the Holy Spirit has put on me to pay attention to is the wording. And when I started to do some research on this, I could feel it in my spirit not to look at anything within the last year or so, year and a half. Because if you think about it, all the information that we're going to see in this past year and a half is definitely going to be skewed, manipulated to work out the way that it's supposed to. And I got to be very, very careful with my wording here, you guys. Now, if you look at the screen here, even the word translation. So watch the video after this and you'll know what I'm talking about. This is talking about mRNA and its main function and responsibilities. And one of the processes is called translation. Where have you heard the word translation before, you guys? Where in the good book have you heard the word translation? Well, I'll tell you. Enoch. When Enoch was translated, he was translated from what? From the flesh to spirit. And he didn't die. Now, I believe the wording says a lot. And again, praying on it, was told to pay attention to the wording. On the right-hand side, what do you see there? Anticodons. Coders. They're mRNA is responsible for pulling apart and then recoding to the way that they want it. What's in the code, you guys? We all have a pretty good idea of what's in the code now at this point. What exactly it is, will we ever know? I don't think it really even matters, to be honest with you. So if you see the word there, anticodon, even the word anti, and there's another a little earlier in the video, you'll see they explain it anti as well. Now, nothing is by accident, brothers and sisters. You all know that. And I encourage you, and I'm told to tell you, encourage you to do research. And when you do your research, go back from when they were first starting to figure it out or really starting to master it. So 10 years back, go as far back as you can find the research, because this was when they're in their first initial stages of understanding of what it was, and they're all excited to publish the papers and show the rest of the world. Well, now that they know what's going on, and now that what's supposed to be happening is happening, and again, I know I'm speaking in riddles right now, but I got to be careful. That information is not going to be the same as it was in the past. And again, we're supposed to take all of this to the Father, all with a grain of salt. And you're probably sitting here going, okay, what are the purposes of these videos? The purpose is so that we can explain to fellow brothers and sisters that still have scales on their eyes what is happening. And to go along with the other 2 or 3 videos that I've posted in the last 24 hours or so, it's a great opportunity for you to show them there's something going on here. When we try to speak to somebody in the spirit and show them what's happening in the spirit, they cannot hear it. They do not have the ears to hear it or the eyes to see it. Therefore, they will not. So all they're going to read is your emotion. They're going to pick up on how excited you are about it and what you're trying to explain, your zeal for it. And sometimes it put people off because they don't know what you're trying to tell them. And so therefore they're not able to understand it. Now, this is just one more tool that you can use so that you can show brothers and sisters, look at like what is in the code. That's the main question. What and I know you're going to get that the answer is, oh well, it's the code that's necessary so that things go back to normal. Okay. Now there's videos out there, countless videos out there of even Mr. Gates himself saying, if we do a good enough job with this, what goes in our arm, we can reduce the population. Now, that's not a Freudian slip, you guys. That is the truth, hiding the truth in plain sight. So those videos are easy to access to be able to show somebody. Now, at the end of the day, you're going to have your brothers and sisters that just don't want to hear it. That's okay as well. As long as we're doing our part, dear brothers and sisters, and that's trying to pick up your cross daily, is to do the best with what you have. And this is what I'm trying to do with these videos here, is just to give you one more avenue, one more tool, and that tool bag of yours to be able to explain or try to explain the science behind it. Because a lot of them, that's all they trust right now. Trust in the science. That's it. That is a phrase for a reason. Okay, well, if you want to trust in the science, well, here's the science. Here's a breakdown of your beautiful strand of DNA being torn apart and recoded with anticodons, and now you will be translated into something different. This is fact. This isn't made up. This is their words, not my words. To take who you are, pull you apart, and then recode you with something else is exactly the purpose of mRNA. So I'm going to leave this here. I'm going to attach the video that I want you to watch that's on the screen right now. I pray you get something out of it. It's just a beautiful opportunity for you to share with people that are coming to it. And without further ado, I'm going to be quiet and let her go. I just want you to know I love you all. May God bless and protect all of you. And let's keep sounding the alarm, you guys. I love you. Goodbye.
In this video, we're going to talk about transcription and translation. And here is just a basic overview of these two processes. Transcription is the process by which DNA gets converted into mRNA, also known as messenger RNA. And translation is the process of converting the information stored in messenger RNA and using it to build protein. Now, if you recall, which organ now is used to make proteins? What would you say? The answer is the ribosome. The ribosome is the site at which proteins are manufactured in the cell. So we're going to talk about that shortly. But let's go over transcription first before we talk about translation. Transcription occurs in 3 steps: initiation, elongation, and termination. Now, during initiation, RNA polymerase binds to the promoter region of DNA. Now, what is the promoter region? The promoter region is basically a short sequence of DNA. In eukaryotic DNA, it's TA, TA, AA, also known as the Theta box. Now, this sequence is located 25 nucleotides upstream of the site where transcription begins. The next thing that RNA polymerase does is it causes the 2 DNA strands to separate. And during elongation, it begins to add nucleotides to the growing mRNA strand that we see here. Thus, RNA polymerase synthesizes mRNA starting from the 5' end going to the 3' end. However, RNA polymerase, it reads the DNA strand in the 3 to the 5' direction. Now there's 2 strands that you need to be familiar with. The first one, the one that's used to synthesize mRNA, is called the template strand, or the antisense strand. So that's the one in which RNA polymerase is active upon. The other one, which is not used, this is called the nontemplate strand, also known as the sense strand. Some textbooks will call it the coding strand because its sequence matches up with RNA, except the fact that uracil is found in RNA, but thymine is found in DNA. Now, during the last step of transcription, which is the termination step, the RNA polymerase molecule, the mRNA strand, they all separate from the DNA template strand. Now, in this step, the poly-A polymerase enzyme, it caps the 3' end of the mRNA strand, and this is known as the poly-A tail. Now, also during the beginning of transcription, the 5' end is also capped, and the reason for this is to protect the mRNA strand from being degraded by certain enzymes. At the end of the termination step during transcription, DNA has been used to create a pre-messenger RNA strand. Now, this particular strand has something known as introns and exons. Introns are basically longer sequences of nucleotides that do not code for anything. So they must be removed in a process known as RNA splicing. Now the exons, those are shorter sequences of nucleotides, and they're going to be used to synthesize proteins. And so the exons remain, but the introns, they must be removed. And so now we have a completed messenger RNA strand. Now let's work on a practice problem. Let's say if you're given a sequence of nucleotides on a DNA strand, and you're asked to write the corresponding sequence on an mRNA strand. What would it be? Feel free to pause the video and try it. So if we're reading the DNA strand from the 3 to 5 direction, we're going to have to write the corresponding mRNA sequence in the 5 to 3 direction. So what letter corresponds to G? It's important to know that G always corresponds to C, and vice versa. C corresponds to G. Now what letter corresponds to A? A usually corresponds to T, but there's no T in RNA. Instead, A is going to correspond to U for uracil. But T in DNA corresponds to A in RNA. And so if we continue, everything else is going to be U, C, A, U, A, U, G, C. And so this is the mRNA strand that corresponds to the nucleotide sequence in DNA listed above. Now let's talk about translation, which is the process of taking the information stored on an mRNA strand and using it to construct a protein. So once the mRNA strand is synthesized in the nucleus, it leaves the nucleus and enters the cytosol, where it interacts with a free ribosome or one that is attached to the rough ER. Now within the ribosome, it's going to interact with a tRNA molecule or a transfer RNA molecule. Now let's focus on the sequence of nucleotides on the mRNA strand. Notice that they're separated in sets of 3. Each set of 3 nucleotides represents a codon, which matches up with another 3 nucleotides on a tRNA molecule known as an anticodon. And each codon matches up with a specific amino acid. And so that's how the information stored in the mRNA strand can be used to construct a specific protein. And we're going to talk more about this later in this video. Translation, like transcription, occurs in 3 steps. initiation, elongation, and termination. So here in this picture, we have a ribosome, which is composed of two subunits, the small subunit and the large subunit. And the ribosome has three active sites, the E site, the P site, and the A site. Now, during initiation, we have the start codon AUG, which corresponds to the anticodon UAC. Now, that particular tRNA molecule has the methionine amino acid attached to it. And this tRNA molecule, it enters the ribosome at the P site, also known as the peptidyl site, where the peptide bonds are formed. Now, during the second step of translation, that is during elongation, another tRNA molecule enters the A site. So I'm just going to draw it here. And during that process, a covalent bond will form between the two amino acids that we see here, highlighted by the red circles. Now, as the process continues, the tRNA molecule in the P site will move to the E site. And as it does so, it's going to lose an amino acid. And so what's going to happen is we're going to have a growing chain of amino acids that will leave or extend out of the ribosome. So make sure you understand that the tRNA molecules, they enter the A site and then they exit from the E site. And in the process, the amino acids are being joined together. So over time, this polypeptide chain is going to grow, creating a protein. The last step of translation, the termination step, begins when a stop codon is read. Now, there are three stop codons that you need to be familiar with. The first one is UAA, and then the second one is UAG, and the third one is UGA. Now, these stop codons, also known as nonsense codons, they don't code for any specific tRNA molecule. Instead, when these are read at the A site, they cause a release factor to enter the A site, which causes the small and the large ribosomal subunits to basically disassemble. So this is where translation ends, and the protein, it leaves the ribosome, where it eventually goes to the Golgi body for further processing and modification. At the Golgi body, the proteins undergo folding, where they form a specific shape to perform a specific function. And that's basically it for this video. Hopefully it gave you a good overview of transcription and translation. So that's all I got. Thanks for watching.